We present our paper, Regional Brain Volume Changes in Hakim Disease versus Alzheimer's and Mild Cognitive Impairment. My name is Shigeki Yamada, and I'm from the Nagoya City University. We disclose our potential competing interests. First, we will explain the name Hakim disease. This name was proposed by the task force team for renaming chronic hydrocephalus in adults within the International Society for Hydrocephalus and Cerebrospinal Fluid Disorder. It referred to the condition previously known as idiopathic normal pressure hydrocephalus. The term idiopathic implies an unknown cause. However, as our understanding of the disease has advanced, it's no longer entirely cause unknown. Additionally, normal pressure indicates that intracranial pressure is within the normal range. However, this is not always the cause for all patients. We propose the name Hakim disease. We recognize as one of the three major diseases in the elderly, alongside Alzheimer's disease, which represents cognitive impairment, and Parkinson's disease, which represents movement disorder. Hakim disease presents with both cognitive and motor impairment. Its prevalence and available treatment options are between those of Alzheimer's and Parkinson's disease. In 2023, Fujifilm first released this AI-based brain segmentation application, which can automatically segment the brain into 21 subregions and CSF into 5 subregions from 3D T1-weighted MRI. In 2024, the new version can segment the brain into 100 subregions and CSF into 7 subregions. It allows for precise extraction even in case of Hakim disease, which involves significant ventricular enlargement and brain deformation. In this study, we use this new version to analyze the distribution of brain and CSF subregions across different diseases. The study population in this study includes the following conditions: Hakim disease, Alzheimer's disease, Hakim plus Alzheimer's disease, mild cognitive impairment, and normal volunteer. The cortical and subcortical gray matter volume ratio decreased linearly with aging, whereas the cerebral white matter volume ratio slightly increased until the 40s and then declined after the 50s. The most pronounced reduction in volume ratio in Alzheimer's disease was observed in white matter. As age increased, the subcortical space volume ratio compensatory increased, which was higher in AD and MCI group but lower in HD. The volume ratio of ventricle increased in both HD and HD plus AD group. HD and HD plus AD groups exhibited significantly lower volume ratio in the supramarginal gyrus, pars triangularis, paracentral gyrus, inferior cortex compared to the other three groups. AD group had a significantly reduced volume ratio in the hippocampus and temporal lobe, particularly in the entorhinal cortex, fusiform gyrus, and inferior temporal gyrus, which are markedly smaller than those in the other four groups. These cross-sectional images show the hippocampus, surrounding temporal lobe, midbrain, and the inferior horn of the lateral ventricle. Mean Grassi delta was used to compare the average volume ratio of segmented region between healthy control aged more than 50 years and each disease group. The AD group exhibited the most significant volume reduction in the hippocampus, entorhinal cortex, and temporal lobe. The inferior horn of the lateral ventricle was notably enlarged in the HD, HD plus AD, and AD groups. The surface views of the mean Grassi delta in Hakim disease exhibit significant volume loss in the supramarginal gyrus and the surrounding parietal lobe. Alzheimer's disease exhibit much more pronounced volume loss in the hippocampus and temporal lobe. HD and AD group exhibit much more pronounced volume loss in the supramarginal gyrus and the surrounding parietal lobe. However, unlike the AD group, no volume loss was observed in the temporal lobe. MCI group exhibited volume loss across whole brain but did not show distinct reduction in the hippocampus and the temporal lobe, as observed in the AD group. These are our conclusions. Finally, we thank the radiology staff of our collaborative hospitals and acknowledge the support of research grants.